So why we need to study biotechnology in detail in the grade 12? Why? Because we all know very well in the modern industry to give you a classic medicine at a very cheap price with a very good quality and quantity is satisfied only with the help of the biotechnology tools. We know very well diabetic patients are suffering with insulin synthesis. That is the reason why they can't reduce their blood glucose level. So we are giving the insulin at a cheap price after getting the insulin synthesis from a genetically modified microorganism. That is why we are studying the principle of biotechnology, not only to solve the medical issues, not only to solve the genetic disorders, not only to give the grains at a very cheap price for serving so many number of activities. We are using principle of biotechnology. Each and every segment of the biotechnology is having a cluster of genetic tool. We are using enzymes as a molecular tool to break the DNA, to break the protein, to attach the DNA, to attach the DNA, and allow and to attach the broken RNA. So we are using enzymes as a tool. We are using microorganism as a tool. We are using microorganism DNA as a tool. So all these things we are using as a tool in the case of principle of the biotechnology chapter, right? So now we are entering into this chapter in detail. So any one of you are having any doubt, please ask me. I'm there with you to help you, right? So in this chapter, principle of biotechnology, what we need to understand basically, as I already told you in the introduction segment, before commencement of this particular lecture, here microorganisms are modified genetically, right? Microorganisms are modified genetically in the sense to basically to give you valuable products. At the same time, we should not disturb the existing living structure by producing the side effect imparting molecules. For example, I am using microorganism for getting the valuable components. Along with that valuable component, if some other unnecessary components are getting synthesized, then it is a great issue for us. That is the reason why we are choosing a genetically engineered microorganism to get your valuable components, plus the side effect importing components should be removed, should be nullified in terms of its activity. European Federation of Biotechnology, they given a classic definition for the biotechnology, incorporates both traditional and modern molecular aspects, right? So as I already told you, we are using biotechnology not only for the medicine, not only for the agriculture, not only for the industries, not only for the home products. Okay, we are using biotechnology for nullifying the harmful microorganism activity also, which is coming much more in the case of bioweapon war, right? So now you know very well in the principle of biotechnology chapter, the genetic engineering molecule, whatever we are using here is not only DNA, RNA also we are using. So introduction of the modified genetic material. What do you mean about introduction of the modified genetic material? So we are introducing the desirable gene into the respective host by incorporating the desirable gene into the host DNA. Again, back I'm repeating. For example, let us consider the desirable gene is A. So with the help of the microorganism DNA, I am introducing the A gene into the host DNA. So into the host DNA, I am incorporating. In the host DNA, I am incorporating the desirable gene A by using microbe. The above one is M, I am writing. Microorganism DNA is used to incorporate desirable gene A into the DNA of the host of a specific target organism. Why, sir? We need to introduce a specific gene into the target cell DNA. Why? Because we are making the target cell DNA to be more effective from the performance category. So the performance category of a cell should go up. For that, we are introducing the desirable gene into the target cell or host DNA into the target cell DNA or host DNA, to which we are introducing the desirable gene with the help of microorganism DNA. By using microorganism DNA, I am introducing the target gene A into the target DNA or host DNA. And I'm making the host DNA to synthesize the valuable components by using the A gene. So that is the main advantage of the genetic engineering, which is a part of the biotechnology, right? So if you're talking about the bioprocess engineering, you know very well the bioprocess engineering is one of the subject in BTech biotechnology and biotechnology. It is coming under the BS honors degree. So BS honors biotechnology, BE honors, BE biotechnology. In all these courses, there is one separate paper is there called bioprocess engineering. That bioprocess engineering is one separate paper. That paper they given as a paragraph in your CBSE textbook. We need to understand why we need to do the bioprocess engineering. Bioprocess engineering, we are creating and maintaining a sterile environment. For what for we need to make a sterile environment? Why? Because to stop the other microorganism growth. For example, I like only A microorganism to grow, but not B and C microorganism to grow. Accordingly, I am maintaining the sterile condition in the sense the specific pH, specific temperature is maintained. And next important point, what we need to understand is free from microorganism contamination. So the bioprocess engineering should have free from microorganism contamination in chemical engineering process. The controlled environment is essential for the growth of specific organism in a large quantity. So basically the genetically engineered microbes, we are growing in a controlled pH, we are growing in a controlled temperature. Why? Because to emphasize the high quality antibody production, high quality vaccine production, high quality enzyme production, and much more. So, for producing the huge concentration of the antibody, for the production of huge amount of antibiotic, for the production of vaccine enzyme at a very less price by using the microorganism DNA, we are using different type of microbial DNA. By using microorganism DNA, I am getting the valuable human missing enzyme. For example, one particular human patient is having a defective enzyme. The defective enzyme I am making with the help of the microorganism after modification. So there is nothing but bioprocess engineering. So in this bioprocess engineering, we are making antibiotic vaccines, right? Okay, so how we are using microorganism? Certain toxic genes will be there in the microorganism DNA that I am removing it. After removing the toxic gene of the host DNA, after removing the toxic gene of the host DNA, I am removing the toxic gene of the host DNA by using a specific enzyme, and I'm incorporating a specific desirable gene into the host DNA and making the host DNA to be more viable in terms of its activity. For example, I'm host DNA, I need to maintain cell division. I'm the host DNA, I need to maintain cell multiplication. I'm the host DNA, I need to make the cell components. I'm the host DNA, I need to make the cell performance depending upon the situation. So like this, I'm having so many maintenance function. So that maintenance function sometimes I'm incapable to do. That is the reason why the scientists are introducing the desirable gene into the host DNA, right? So now we are entering into the next important topic in the same principle of biotechnology, how we can introduce the DNA into the host DNA, into the host DNA. So if you're seeing this picture, if you're seeing this picture, I think so, you can see the picture here. This is nothing but a host DNA, which is nothing but a host DNA, which is nothing but a host DNA. Can you see this picture here? The host DNA is not given here. Okay. So, for example, this is the host DNA. This is the host DNA. I drawn here. Okay. To this host DNA, the foreign gene is given by the bacterial DNA. The bacterial DNA is having the desirable gene of mine. So, once again, back I'll repeat. This is the plasmid DNA of the bacteria. The bacteria cytoplasmic DNA or plasmid DNA, whatever I am writing in this place, this plasmid DNA, which is nothing but a cerebroplasmid DNA, and that cerebroplasmid DNA or plasmid DNA is used for introducing the residual gene. What gene, residual gene we are introducing? So this is the foreign gene, which is a residual gene. This might be an insulin gene, might be a hormone gene, whatever the growth factor gene, anything I am introducing into the plasmid. So this red color small DNA fragment is a gene which I am introducing into the plasmid, and I am making the plasmid to introduce the visible gene A to a host DNA. The host DNA is getting this desirable gene. How come the host DNA got? Because who has introduced? The bacterial plasmid DNA has introduced. To the bacterial plasmid DNA, who has introduced the foreign gene? Okay, the scientist we introduced. By using what we introduced? By using DNA ligase enzyme. So what we need to understand basically, by using DNA ligase enzyme, and by using the restriction enzyme, we introduce a foreign gene into the plasmid, and to this plasmid is used for introducing foreign gene into the host DNA, right? So this is the way we need to remember. Now we are entering into the principle of biotechnology in detail. In this particular chapter, what we need to understand, the alien organism having a what DNA, piece of DNA is used to introduce this piece of DNA into the host DNA. In the host DNA, we are introducing the required DNA by using host. Into the host DNA, I am introducing the residual gene by using vector. Now we are coming for the next segment. The alien DNA becomes integrated into the host genome. Alien DNA is getting integrated in the host genome. Okay, by using what exactly? So the alien DNA become integrated into the host DNA or host genomic material with the help of the two important enzyme. One is called as what? Restriction enzyme. Another is called DNA ligase enzyme. By using restriction enzyme and by using the DNA ligase enzyme, I'm introducing the foreign gene into the host DNA. So this is the origin of replication. The origin of replication. So origin of replication is a segment where we can decide. Okay, we can design this much amount of DNA duplication is possible with the help of a specific DNA, right? So what we need to understand basically, the origin of replication or ORI.C are nothing but a replication initiation point, okay, which is very, very important in the alien DNA. Why? Because if the origin of replication is weak, I can't introduce the big size gene. If it is strong and can accommodate the big size gene, right? So this alien DNA or foreign DNA can multiply the host DNA, known as cloning or making the multiple copies of the template DNA. Second back I'm repeating, it's very, very easy to understand. Here, if you're talking about the host DNA, might be my skin cell, might be my brain cells. My skin cell is not whitish, let us consider. So to this skin cell DNA, I introduce a foreign gene by using the microorganism. To the microorganism, I introduce a foreign gene by using restriction enzyme and DNA ligase enzyme. So by using restriction enzyme and DNA ligase enzyme, I introduce a foreign gene of my interest into the host DNA, right? Now, Salmonella typhi is a bacteria which is having the different DNA incorporation. Okay, so what we are doing is the first recombinant DNA involved linking a gene encoding antibiotic resistant. So the gene which makes the enzyme, that enzyme will not make the antibiotic to survive. So that antibiotic making enzyme which is made by the gene, that gene I'm introducing into the plasmid of the Salmonella typhimurium. Salmonella typhimurium is a bacteria. This bacteria, again I'm repeating, Salmonella typhimurium is a bacteria These bacteria having the cytoplasmic DNA, the cytoplasmic DNA of Salmonella typhimurium is incorporated with the foreign gene. So Salmonella typhi, I am writing as S.T, and this bacteria plasmid is having what? Foreign gene of interest that we can introduce into the Salmonella typhi is a bacteria. Okay, that bacteria is having the cytoplasmic DNA or plasmid DNA into which we introduce a foreign gene. The foreign gene is introducing into the host. The foreign gene is introduced into the host DNA. The foreign DNA is introduced into the host DNA. This is the way we are making the recombinant DNA. This is the way we are making the recombinant DNA. Are you getting my point now? So what we need to understand is molecular scissors. What do you mean about molecular scissors? Molecular scissors are nothing but restriction enzyme which makes the DNA fragmentation to happen. So because of the DNA fragment, uh, whatever you are getting because of the molecular scissor is nothing but the restriction fragment. Why? Because we got those DNA fragments by using restriction enzyme. By using restriction enzyme, we got the DNA fragment. Next point, what we need to understand is, for example, if I need to introduce the foreign gene of my interest in the host DNA. If I need to introduce foreign gene of my interest in the host DNA, I'm using an enzyme called DNA ligase enzyme. By using DNA ligase enzyme, I am introducing foreign gene of my interest in the host DNA. So the recombinant DNA was transferred to E. coli, a closely related bacteria to the Salmonella, where it could what replicate. Okay, who will be replicating? The recombinant DNA of the E. coli is transferred into the Salmonella typhi. Okay, and in the Salmonella typhi, this DNA will multiply, right? So what we did basically, we made the E. coli DNA and we linked it with the Salmonella typhi DNA. So Salmonella typhi DNA and E. coli DNA are combined in such a way, the E. coli and Salmonella hybrid DNA can multiply in E. coli as well as in Salmonella. So how we bridge this DNA of two different species by using DNA ligase enzyme. So this DNA ligase enzyme I underlined by using DNA ligase enzyme, I attached the two different species DNA. Now what we need to understand is these steps are fundamental to the genetic engineering and biotechnology, right? So we are introducing the host gene into the host. No, we are introducing the interested gene into the host DNA by using the microorganism. So the one copy of DNA runs 20 cycle of PCR. That means number of copies equal to initial copy into 2 to the power of n number of cycles, right? So here 1 copy into the n we are substituting with 20. So 1 into, okay, 2 to the power of 20 will be giving this much amount of copies. So, so if you're running the DNA, okay, this formula always you need to keep, okay, initial copy we are keeping as 1 and then 2n, 2 to the power of, okay, number of cycles. So this is the way we need to do the mathematical biology question in the principle of biotechnology chapter. Now we are entering into gel electrophoresis analysis. In the gel electrophoresis analysis, we know very well the two DNA fragments are separated, one with 500 base pair, another with 800 base pair. If they travel through the gel for the same duration, which fragment will be closer to the positive electrode? Means obviously the smaller DNA fragment will travel further and be very closer to the positive electrode doing the gel electrophoresis. Okay, smaller DNA fragment, Farad MB will travel further, okay, far away. Okay, we can say another way around them and be closer to the positive electrode. Okay, doing gel electrophoresis. So the DNA fragment is having the negative charge. Okay, 800 base pair is also having the negative charge, right? So doing the gel electrophoresis, Obviously, which fragment is very closer means obviously the 500 base pair DNA is very closer. Okay, if you're talking about only the charges, 800 DNA will be first, but we are not talking about only the charges, we are thinking about gel porosity also. So gel porosity and the number of negative charge of the particular DNA fragment will make the DNA to get migrated towards the respective electrodes. Towards the respective electrodes. Now we are entering into plasmid transformation efficiency. If you transform 1 microgram, the mu stands for milligram or microgram. Microgram. 1 microgram of the plasmid, 1 microgram of the plasmid, if it enters into the bacteria, if it enters in the bacteria and requires 100 cycles, what is the transformation efficiency in the colony formed unit area? Okay, they are asking microgram specifically. This microgram only represented with the mu. Okay, nanogram with represented with the small ng. So be very careful. So what we are saying, okay, how we are saying in the plasmid transformation efficiency, if you transform 1 microgram of plasmid into bacterial DNA and recover 100 colonies, what is the transformation efficiency in colony making unit per microgram? Okay, here we know very well the number of colony forming units into one into 1 microgram into 1 microgram. So 100 CFU, 100 CFU. This is the answer. I think so. You understood this message. So these are all the mathematical biology questions asked exclusively from the CBSE hard questions portal, and this high order thinking skill questions will make you a little bit to think more, so that what you'll be lagging in terms of finishing the answer script. So this is the way they will filter you. So be very familiar with the mathematical biology, right? Calculate the growth rate. So in this particular calculating the growth rate, the culture of the bacteria grows from 10 to the power of 6 cells to 10 to the power of 9 cells in 5 hours. Calculate the growth rate. Growth rate equal to final cell count minus initial cell count. Okay. The values they already had given. So what we are subtracting the 10 to the power of 6 in 10 to the power of 9, so that you are getting what 10 to the power of 6 9 divided by 10 to the power of 6 divided by 10 to the power of 6 into 5 hours. So the answer is 1 lakh, 1 crore, 98 lakhs. These number of cells are there in 1 hour. So this is the way they will ask you to calculate the growth rate. I think so. Any one of you are having any doubt? So no one has the doubt. So now I give the provision for you to ask the doubt. Okay, these are the worked examples questions we discussed in detail. And then we discussed about the related videos. Now we are going for tips of the exam. All the terms we need to remember in detail. What are the terms we used as of now? Okay, exonuclease enzyme, endonuclease enzyme, DNA ligase enzyme, plasmid DNA, host DNA. Okay, we use restriction enzyme, we use electrophoresis. All these terms we need to remember in our mind, and we need to bring the appropriate concept for those terms, and then we need to recall, and then we need to say whether we are correct, we are correct or not, like this, right? This is the tips for the exam, right? In the tips of the exam, we need to study what is in the restriction enzyme. Restriction enzyme, how many types of restriction enzymes are there in the world? Majorly 2 types, but furthermore it is divided into many based on the species name. Based on the species name. So based on the type of subspecies, many subvarieties of restriction enzymes are there. Many subvarieties of restriction enzymes are there. Again, I am repeating, many subvarieties of the restriction enzymes. Many subvarieties of the restriction enzymes are there. So, what restriction enzyme we need to use depends upon the length of the gene, whatever you are choosing, to incorporate into the respective host DNA. The host DNA, this much lengthy gene I am going to introduce, then you use that particular specific restriction enzyme. No, I am introducing only small size gene, accordingly you choose a specific restriction enzyme, right?